and we'll turn on the music. From Microbe TV, this is TWiEVO. This Week in Evolution, episode 115, recorded on June 18th, 2025. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Hey, Vincent, great to be with you from Elde Lab Studios in sunny, hot, and dry Salt Lake.
It's the opposite of here. It's been raining forever.
Hmm.
Today it's a little warmer. It's cloudy. Thunderstorms expected. It's 26 C. And the humidity is, you have to scroll all the way down to this stinking thing. Where's the humidity? 78%.
Ooh, that sounds pretty tropical.
Out there it's dry right now. Where you are?
It's dry. And honestly, we haven't had a drop of rain for going on a month now. There was a big storm system that was supposed to come through, but got pushed to the east into Colorado. I guess the only forgivable sin here is it wasn't during ski season, so we didn't lose powder in the exchange. But yeah, it's dry, and we're looking for a little precipitation. So yeah, send it down our way. We'll take it.
You don't want to lose powder, do you?
You don't want to lose powder. That's sort of a top priority.
That'd be like tragic. Welcome, everybody, to TWiEVO. We have our moderators here today. Let's thank them.
Yes.
My viewers' number is not working, so I don't know who's here.
That's okay.
Maybe there's no one here. We got
just us.
We got Barb Mack. Thank you, Barb Mack from the UK, where it is late, right?
Right.
Well, 1 p.m. This was 6 hours ahead. That's like 7 or 8 p.m. That's not too bad.
Yep. Could be worse.
The worst one is the office hours at 8 p.m.
Ooh,
Thank you, Barb Mack. We have Les from California.
Fantastic.
Tom Steinberg is here from Western Oregon, where they need rain.
Yeah, where the West is a little dry and toasty.
Here we go. 17 people now. It's updating. So tell us where you are from, folks.
Yeah, please do. We're a little slow out of the gates here, a couple minutes behind schedule on the live stream. But that's just us behind the scenes working out the kinks and firing up the engines for TWiEVO 115, Vincent. This is...
yeah, look at this. Now, we got the wrong date here. Les just pointed this out to me.
Those are the Easter eggs that we're putting in for
I don't know why. I do that every often. I make so I have a template for the thumbnail, and I just modify it. And what am I thinking? I'm just
a lot of details, and we don't run the tightest ship in the shipping business.
Yeah, we don't. It's all easy stuff. It's trivial compared to what I used to do in science, but
yeah,
I don't get it. Hey, folks, tell us where you're from. We'd love to announce that here.
That's right. And open the dialogue for the science ahead as well. We have a really interesting, I think, provocative story that will be kicking around.
Very cool story. Yes.
Yeah. For TWiEVO.
Look who's here.
There we go. That's a familiar face. And I have to say, Vincent, so good to have Bob Elde, my father, along for the ride. The paper we're talking about, the preprint, he alerted it to me a couple days ago. So we take tips from family and friends on TWiEVO to think about the science that's worth.
Why does he say maybe? Maybe, do you remember what it is that we said? Anyway, it doesn't matter. Bob says it's 21C on Marrowstone Island. What's Marrowstone Island?
Yeah, so that's on the Olympic Peninsula up in the Pacific Northwest, near Port Townsend, Washington, about a half hour from there. Great little spot, kind of hidden in plain sight across from Seattle.
So Les said, loved seeing the Elde Museum.
Oh, fantastic.
That was in Johnson City, Texas, right?
Correct. Yeah, exactly. So in his illustrious retirement, Bob Elde is co-founder and science director at the Science Mill, which has some new leadership. Vincent, there's a new CEO that's taken the reins of the Science Mill, some fresh updates of the exhibits and related items. And so it's a good time at the Science Mill. Anyone in the Texas, sort of San Antonio, Austin, Hill Country area, if you haven't been to the Science Mill yet, or it's been a little while, great time to check it out.
Tom recently heard Harmeet at UR recently.
Oh, fantastic.
Dual host viruses, fitness trade-off, and fitness plateaus. Sounds familiar, right?
Yeah, that's a really cool story out of the Mullick Lab. Speaking of Seattle, we're circling around to the Pacific Northwest again. Glad you made it, Tom. That's great. Yeah, so just for everyone out there, Harmeet was my postdoc mentor at the Fred Hutch Cancer Center in Seattle. Let's see, I was there, Vincent, from 2005 to 2011 before I migrated east to Salt Lake. And at the University of Utah, a place we sometimes refer to affectionately as Fred Hutch East. In addition to me, there's a number of former postdocs now running labs here, sort of a gold rush from Seattle to Salt Lake for science.
Yeah, I always enjoy Harmeet. A lot of fun to talk to.
Yeah.
Go ahead. Sorry. Go ahead.
Oh, sorry. I was just going to say I'm going to see him next week. So I'll be out on the East Coast at Stonehill College. There's a Gordon Research Conference starting on Sunday, I guess already, called Molecular Mechanisms of Evolution. And Harmeet is one of the keynote speakers. And so it's been a minute since we've been together. I'm looking forward to catching up with him and many others, including, I don't know, I kind of don't mean to sort of break the news here, but we have a new colleague coming to Salt Lake, to the University of Utah, a fellow called Mohammed Siddiq, who's a postdoc at the University of Michigan, and he'll be joining my department here in human genetics at Utah. He's coming this fall. And so I'm looking forward to catching up with him and many others at the Gordon Conference near Boston. Yeah.
Abdulaziz is in KSA, 40 degrees Celsius. That's pretty hot.
Jeez, that is toasty.
Very toasty.
Yeah.
You need to be inside.
Indeed.
Jeremy's in Seattle. Welcome, Jeremy.
Great to have you here.
So Bar Max, it's 1800 there, which is 6 p.m. So not so bad.
Not so bad. Office hours, that's another story. Kind of.
Hello. You have to be... sorry. I keep interrupting.
No. You have to be nocturnal to catch office hours if you're over in Europe, I would say.
But we have the guy here who comes here. He's from Norway, Dagfinn. Mm-hmm. Sometimes he shows up at office hours too. It's 2 a.m.
Love it.
Vanity Nutrition has joined a moderator. Welcome, Vanity.
Fantastic.
Nice
welcome.
To have you.
Yeah.
You got your feet on the ground. Vanity is often 38,000 feet up.
Mm-hmm.
Pete is in London. 6 p.m. Still like summer. A week of fine weather expected. 27 to 30 C. Thank you for the weather report.
Yeah, and good to see you, Pete.
Did you hear my message?
Oh, wow.
I don't know. I'm guilty as charged. You know, I have.
Yeah. Cliffhanger here.
I'm often, sorry, I didn't. Where is the message? And I will go look for it, Vanity. I'm very sorry. Noir LeBlanc is here in Santa Fe. Welcome.
Oh, very cool.
Welcome. 76F, 6,700 feet altitude.
Yeah, I'm weighing in at about a little under 5,000 feet. So you've got the higher ground, literally speaking.
Let's see who else we have here. FSM is the dog is from Wales. It's 23 and threatening 30 tomorrow.
It's warming up a little bit.
Bad Cat is in Winthrop, Maine. First time watching TWiEVO live. Bad Cat's often on the office hours.
Oh, very cool.
Cross-pollination.
There you go.
This is not the Gregor Mendel episode, folks. So, you know.
That's right. We were talking about the pea plants last time. We're going to go into the microbial world here in a minute or two. Really interesting evolutionary sighting on tap for TWiEVO 115.
Gary Weiner's in Denver.
Great.
Science Insanity. Yeah, that's actually what we do here. If you like these
Yeah.
programs, we'd love your support, right? You can do a tip here. There's called a super chat, but even better, go to microbe.tv/contribute. And there are other ways you can contribute as well. We'd love your support. We don't do ads on these programs, so to keep it focused, right?
We don't do ads, but we aim for sanity along with our science.
It's good. I like that, Gary. Get back for some science and sanity.
Yeah, that's
Although,
great.
No one ever accused us of being sane, Nels. I don't know. At least
Not yet.
scientifically sane.
That's right.
Red Desk. It's great to see you smiling in the face of the current political climate. London 28C. Well, yeah, but we're very hurting inside. I'm telling you, this is horrible what is going on.
Yeah.
And, the American voters, if the under 30s had just voted, this would be very different. But they stayed home because they don't like the parties. Okay, but now you look what we got. We got the worst possible thing ever. Apathetic under 30s. That's not going to get them out next time, I know.
That's your sales pitch. We're going to have to circle back on that science and sanity part here. No, absolutely. Really strong headwinds, to say the least. Chaos, category 4 chaos continuing, not just in science, but across the board. What a volatile last weekend, to say the least. So as we're kind of going from all points on the map here, Vincent, both in Minnesota and, the assassination, literally, of a local legislator there, and others on that list. I mean, my God, things are just careening out of control. And then with a really sad, kind of pathetic link to Utah politics, with the senior senator of Utah after this terrible tragedy getting on social media and kind of making light of the situation. I mean, this is just the upside down world of this is insanity, honestly. And so, yeah, but there's something about, especially as we, I think, are framing science, thinking about what we do, is continuing to smile, not to forfeit our joy or our excitement for what's possible. Right. And so that's part of our, operation here, is exactly that. Is we're going to keep telling the story. We can't help ourselves when we see interesting science like some of the work we'll be talking about today. I can't hide the smile from my face because it's just,
we love science. Yeah,
it's really fun.
So here's a link to the Science Mill. Bob Elde's founder of the Science Mill. There you go.
Yeah, thanks, Les.
Pete said he saw an interesting evolution reference, but forgot what it was now.
Yeah, send it along if that comes back. And I don't know. I mean, our process of identifying work is pretty hit and miss. So if you suggest something that's really interesting and we don't do it, that's not indictment on the topic or the science. It's just we're kind of mixing and matching a lot of stuff as we go in real time. Yeah.
And Les says, go look at TWiEVO 100. We did it at the Science Mill.
Yeah, that was fun.
You could see what it was like. Bob says it's not the Elde Museum. Bonnie Baskin, my wife, was the driving force. I was her wingman. The real name is the Science Mill.
Yes, let's get the branding correct here. Absolutely true.
Alexander's in Paris, 28C. Nice.
Fantastic. Welcome. Good to have you here, Alexander.
Sharon is in Tasmania.
Wow, fantastic. 3 a.m. Uh-oh. We're already in the middle of the night. Yeah.
I had a stroke 3 weeks ago. I'm sorry. I hope you're doing well. You have to find your glasses.
Yeah. Thanks for joining us.
Thanks for joining us. We appreciate it.
Middle of the night as well. Yeah.
Okay, you left me a voicemail. Okay, I will go check that when this is over. I really apologize. I do have a phone. I never answer phone calls because most of them are from numbers I don't know, right?
Of course.
And so I don't read the messages. But it turns out some of them are important. I'm really messed up in that sense that I should answer. I don't like answering the phone. It's just I never liked calling people when that's all we had.
Yep.
And although once I got on the line with someone, it was okay, but just this initial calling was hard. And
I could
that's
agree.
still here. But I love having email and chat and messaging and all that stuff. That's great because you don't have to actually talk to people right away.
That's right.
Tracy, it's wherever Tracy is, it's 31C.
Great. Thanks for being here.
Welcome.
Sounds warm to say the least. Yeah.
Moira says it's thank you for not doing ads. You're welcome. You know, we did ads in the very beginning of TWiEVO. And they were actually quite lucrative, but the ad agency kicked us out. They said, you're not making enough money for us. So, and that was the end of that. I said, screw the ad business. But really, I don't want ads to interfere with learning. That's the bottom line, right?
Yeah, absolutely.
Okay, that's it for people telling us where they are.
Fantastic.
Let's turn this off here, and then we can we can start talking about this paper, Nels.
Yeah, let's dive right in. So this is a preprint on the bioarchive, and the title of it is "A Cellular Entity Retaining Only Its Replicative Core: Hidden Archaeal Lineage with an Ultra-Reduced Genome." A group out of Japan. So Ryo Harada is the first author. Takuro Nakayama is the senior author. I think they're headquartered in Tsukuba, the University of Tsukuba, Center for Computational Sciences in Ibukari, Japan. And then I think Ryo Harada, now located at the Institute of Comparative Genomics, Dalhousie University, up in Nova Scotia, Canada. So, Vincent, we're returning here to kind of a topic that we've been interested in kind of consistently over the years on TWiEVO, and this is sort of touching on the definition of life and what it takes to be a life form. What that means as we are in the genomic age, connecting that to the content of a genome and the notion of a minimal genome. What's the minimum size of a genome where it's possible to still be a life form? And then and when do we cross over? So when we think about viruses, whether they're alive or not, I think we've talked about this before. My favorite definition, at least, is that the infected cell, a cell infected with a virus, is sort of alive. It's its own kind of entity, but a virus particle sort of just hanging out, on a surface somewhere or wherever is not alive. It needs the host to sort of put itself into motion, right?
It won't do anything without a host, right? So, but that means that a virus is an organism with two phases, right? You have the non-living, the particle, and the infected cell, which is living, and that solves all the problems. But
that's right.
People, when you say virus, most people think of the particle, right? Like this particle here above me on the shelf. But the virus is actually an organism with two phases, the particle and the infected cell.
Yeah. And just this definition of life, you'd think here in 2025, a scientist with all of the sort of decades, hundreds of years of thinking about these kind of questions, that we might have it worked out. And the answer is sort of a definitive no. It's really messy and complicated and sort of blurring the barriers. So TWiEVO 70, we picked this up with the great science author, Carl Zimmer. So, and he had a book a few years back called On Life's Edge. And I was just looking it up last night. I was browsing the TWiEVO catalog, Vincent, and that was one of these rare occasions when we weren't podcasting together. So you were...
that was you on your own, yeah.
Yeah, my sort of solo act.
Nels broke out of the band and he went solo, yeah.
That's right, I went solo. It's hard to kind of coordinate three schedules. Carl Zimmer in particular is a busy fellow, but we pulled it off. And so if you haven't heard it already, have a chance, check it out. TWiEVO 70 on Life's Edge with Carl Zimmer. And another topic that we've talked about is... or sort of related topic here is symbiosis, right? So the difference between being a free living life form and then becoming associated with a host. And this is usually, I'd say the field by and large has approached these kind of questions from a bacterial standpoint when we think about microbes that become symbionts. symbionts. And obviously the very most famous case of this, ancient and successful case of this, is the mitochondria, sort of the basis of what it is to be a eukaryote, or the eukaryotic cell. And the notion that there was some free living critter that then, kind of bacterial in nature, that then was engulfed in an ancient cell, lost a lot of its genome, went on to become the mitochondria, and everything that followed, sort of an evolutionary time. And
That's
I would say the field,
it's an interesting comparator for today. We should talk about what's encoded in a mitochondrial genome when we get to this.
Yeah, good point. And the fact that, some of those genes that encode mitochondrial proteins over this, billions of years of evolutionary time, have moved to the genome of the nucleus. And so that's really sort of an interesting genetic entanglement that's going on there as well. And then, so more recently, the field has really looked, there's some great work by locally, there's a lab here that Colin Dale runs. So this transition from free living bacteria in insect hosts that then sort of transition to symbionts, where they now require the host, they give up a lot of genes, but what do they give back? These metabolic pathways that provide the ability to gain nutrients from the environment that the host, the insect or whatever it is, might not have. And then these kind of symbiotic relationships, are sort of historically framed as beneficial, but can also be, thought of maybe more accurately as addictions. The host becomes addicted to the symbiont becomes addicted to the host, and they move forward. And so that field has really sort of stepped through a lot of bacterial sort of, transitions, et cetera, on the road to something like, where did mitochondria come from, or what's the origin even of eukaryotic cells? Interestingly, I think, at least to my knowledge, very little sort of cross-referencing with what it is to become a virus, or the origins of sort of, and that's where this paper, this preprint, sort of picks up the story in a really interesting way. So like a lot of science, this is just sort of looking around you, stumbling around, it's kind of stumbling in the dark.
Going down a dark alley,
right?
Nels.
Going down a dark alley, Vincent, and finding something interesting and being like, oh my God, what is going on here? And then sort of rethinking, what does sort of life around us mean in sort of fundamental ways here. So here the authors are going to introduce us to a archaean, with a very reduced genome, as the title of the preprint suggests, Succinarium mirabelle, and has the designation of a candidate, or Canidus, meaning that this this critter, organism, animal, whatever you want to call it, animal would be the wrong word, but critter actually somehow works, is only known by its genomic sequence so far. So, sort of like disclaimers here. There's no microscopic evidence for what this entity looks like, genetic entity looks like, et cetera. And so early days. But a really interesting, as we'll kind of step through it and build the story, I think a really interesting sighting that's worth keeping an eye on. If nothing else, I think for me at least, kind of, has me rethinking the notion of symbiosis, and how these sort of massive transitions from free living life form to obligate symbiont might take place, or what's even possible.
So Sukuna archaeum, it's a candidatus, but I wonder if Sukuna sounds Japanese, right? I wonder if it has some,
absolutely right.
I don't know if it's where they got it. We'll get to that, I guess.
Yeah. Sukuna Biko na, a deity of small stature in Japanese mythology. Exactly.
Really?
So that's where, So the Sukuna.
Oh, there it is. Yes.
There. I was just listening. My daughter and I listened to a podcast on the way to school called Super Great Kid Stories. And there's a great Japanese folk tale that we were listening to this morning called Inch Boy, Ishimboshi. That's what I would have called this critter. But anyway, so we'll return to that in a moment. But first, let's just describe what happened here. So the group is doing a lot of metagenomic analysis, and in particular, considering these single cell eukaryotes, dinoflagellate. So this is another exotic single cell eukaryote, pretty well described, obviously relative to a lot of archaea. But what's interesting about dinoflagellates, so marine critters, single cell, they have two flagella swimming around in the ocean, sort of show up from time to time when they produce sort of poisonous toxins that can get into things like shellfish, et cetera. And But what dinoflagellates are also doing is they're hosting little communities, and this is an area of active research. So what is the sort of microbial diversity within this sort of single cell community? And so you've got bacteria of diverse sort of genetic backgrounds that are potentially kind of on that transition zone from being free living, but are associated with the dinoflagellate, sort of nature's testing ground for new symbioses, and come back in a billion years and maybe you have the next mitochondria in some rare case here, some massively successful story. But they were surprised as they were going through their sequencing, looking for kind of the usual suspects of these bacteria. They put together, as they're analyzing the data, they put together a circular genome, or a piece of DNA that was 238,000 base pairs, those DNA letters, the A, T, C's, and G's. And they're suggesting here, still early days, but they're suggesting that that might be the whole genome for this archaea. And that's really small, Vincent. So, that genome size wouldn't even make a pox virus blush. We know of many viruses with larger genomes than this. And in fact, even for archaea, this is like sort of incredibly small. So I think up until now, there was another, so the smallest free living bacterial genome is something like 1.3 megabases. Right. And in the archaeal world, I think there's another one that's about 490 kilobases, so 490,000 base pairs. 1.3 puts you up over 1 million. And so, so archaea, first of all, what are they? So this is sort of this, second or third branch of life. We've talked about this as well on TWiEVO. Check out TWiEVO 91, Meet Your Very Distant Cousins. So there have been a lot of groups in Scandinavia somehow that have been going out with like little robots going down to these deep sea vents, pulling back samples, and in this case, actually seeing the cells under a microscope, the Asgard archaea. And so these discoveries have really kind of revised or updated the tree of life and put the eukaryotic branch, sort of our branch, our closest relatives, squarely within the archaeal branch of life, next to the bacteria, which are sort of on their own branch. And so we've discussed that a few years back. And we even, before that, kind of tree of life ideas on TWiEVO. I really went deep in the catalog here to TWiEVO 37, A Tangled Tree on the Quamens. We talked to another author, science writer back in the day. This one was a co-hosting scenario. We talked to David Quammen, a really fun discussion on his book. I don't know if I mentioned it, but we just hosted David Quammen in vivo here at Utah back in December, November, December. Really fun to see him and catch up on some of his recent work as we've now walked through a pretty massive pandemic, obviously, since we recorded that. Okay, so again, so what's interesting about this now is we got this sort of massively reduced genome, and then what we'll do is kind of follow along with the authors and see sort of what's in that genome, and then and how are they, speculating about what that means for the evolutionary process, sort of some of the fundamentals of cellular life here. And we've talked about symbiosis before on TWiEVO. TWiEVO 74, one-step symbiosis. This was actually taking E. coli, sort of the lab rat of bacteria, and selecting for mutations in one mutational step, getting to something that's no longer a free living life form anymore. That's part of, depends now on its host. And even some other peculiar critters. So TWiEVO 94, a bacteria, a phage, and a selfish element walk into a cryptomonad. Another sort of sighting there. There's another, I don't know what, this was late. Speaking of sort of nocturnal, this is all a lot of late night trolling through the TWiEVO library. I couldn't find, there's another one we did, Vincent. I don't know if you remember this vaguely at all, but there's another symbiotic event where they identified some immunity genes in another kind of simple critter that they are proposing are mediating that relationship. I really love that paper because I think it helps to kind of reframe how we think about immunity. When we think about it sort of almost overwhelmingly just as defenses against things like viruses or pathogenic microbes, but really to sort of reframe that conversation into thinking about the immune system as a way of having a biological conversation with other genetic entities around us, whether it's viruses or free living cells, bacteria, et cetera, and how that gets mediated. And so, of course, we pay, for obvious reasons, biomedical reasons, we pay a lot of attention to the pathogenic scenarios, but very little attention to probably most of the conversations that happen because there's no pathology there. There's sort of these invisible associations, right? And so this preprint sort of falls into that category. It's sort of something that's hiding right under your nose. Of course, dinoflagellates don't get much attention, so they're also sort of hiding out for the most part in the ocean. Okay, so we've got now this sequence, this plasmid, and they in the preprint they show in Figure 1 sort of a circular diagram of it. We should probably say that it's possible that this doesn't sort of describe genetically the whole critter here. So there could be other chromosomes, other plasmids. Oh yeah, thanks for putting it up. So there it is, the genome of Succinarium mirabelle, its length, and then sort of the circular plot, those tick marks that are kind of in this concentric circle that marks out the different sort of predicted genes that are encoded by the genome. And so you're putting it through some computational analysis to ask, are there open reading frames, start codons, so the triplet DNA code to encode for the start of a gene as it's transcribed into RNA and translated into protein, and is that open reading frame long enough so that it could actually string together enough amino acids to make a viable protein?
So what are those squiggly lines in the middle?
Yeah, good question. Let me pull this up. So I think they're also looking at things like the DNA content. So how many C's and G's are there versus A's and T's?
Right.
And so that's sort of a common just sort of metric that you take. So it turns out there's a lot of A's and T's. That's not unusual for kind of tiny microbial things. It also makes it more challenging to study. But so they find basically, 189 proteins. There's these other tRNAs, ribosomal RNAs. This is all involved in the sort of replication process. And what they kind of immediately land on is sort of, so as you categorize, what are these genes, the proteins encoded by these genes, what are their functions? And what they see is that there's a lot of genes that are involved in just in replication. So making more genetic copies of yourself, right? So you have DNA replication so that you replicate the genome. You have all transcriptional machinery. The DNA goes to RNA, and then you have the ribosomal components, the translation machinery. You go from RNA to protein. And what's sort of striking about Succinarium, as you compare that to a lot of the symbionts, bacterial symbionts, is that there aren't really metabolic pathways or the genes that help you, the enzymes that might take sort of a substrate or some molecule that you are exposed to in the environment and then turn that into a chemical form that can be used by the host for energy. That all seems to be missing. And so, this is, again, just sort of all kind of circumstantial evidence, but that's sort of pointing to a critter that is really sort of focused on replication, perhaps more at the expense of the host, or at the very least, sort of, there's no evidence that it's helping the host in a way that we would think about for a lot of the bacterial.
Yeah, because there are no metabolic genes, which would be the way these symbionts typically help their hosts, right?
That's right. And that sort of sets up that sort of addiction pathway, biological addiction, where if a host can replicate better and outcompete, sort of the other microbes in its environment, because it can now take advantage of this resource that the other ones can't because they lack that symbiont. You can see how those sort of relationships might be successful and sort of take over over time.
This one is harder because it's not providing anything. It's taking, most likely. Right?
Yeah. So that's a great question. And, I'd throw a little bit of cold water on this notion that it isn't providing anything. So a lot of these genes are new to science, right? So and we'll talk about this.
Right.
One of the really cool parts of this is that when they predict those 189 proteins, there's actually a group of them that are quite big. So I think there's something like 13 open reading frames here are predicted genes that make proteins that are would or have the potential to make proteins that are over 1,000 amino acids. That's pretty large, and some of them are like as big as like 4,000 amino acids. That's huge for a protein, and that's even more kind of wild or striking when you consider that the whole genome itself is only 238,000 base pairs. And so you're dedicating a lot of kind of genomic real estate, so to speak, to these like handful of large genes that have really never been spotted before in science. You even put them through.
Yeah. That's probably where the help comes from. Some of those, right?
Yeah. It could. Or so the idea they put forward is that those genes might be involved, and it's true. It could very well be involved in getting access to the host. So, and I love that idea, but it doesn't rule out, right? I mean, they just don't have the evidence yet to rule in or out sort of what these functions are. And so I think, a little bit of caution is warranted that those could be proteins that somehow do kind of fit into the mold of how bacterial symbionts work. It gives something to the host or helps the host do this biology. And here we're talking about the host is dinoflagellates. And so not a lot known about them relative to things like insects or, of course, our own close relatives in sort of the animal kingdom. But they're right. So in the sense that there is a lot of this genetic real estate that is devoted to replication. And that is different, actually, than a lot of the bacteria symbionts. And they do sort of that comparison. It's not like night and day difference, but it's sort of enriched, no doubt about it. For some of these. Also, just a quick note, so that the technology here, I think, is part of what makes this story possible. It opens up a pathway to sort of discover or consider this. So this is metagenomics, but it's going at the single cell level. So there's been a huge push in the last 8 to 10 years, something like that, to do single cell genomics. And so a lot of it is being done in tissues, like from our own samples of mammals or mice or to humans, et cetera. And so, sequencing the genome at the single cell level as opposed to in a population. And so some of that technology they're repurposing or purposing to now do this for these small microbial communities associated with dinoflagellates. And that's sort of what kind of from going from that bulk sequencing to these single cells kind of gives you the resolution to kind of tease this out and look at it in the first place. So again, kind of a time-honored theme for TWiEVO, where we're really interested in the old questions about evolution, the origins of life, where are things kind of coming from, the relationships between them, but then sort of cast in this new light of the updates in technology that make it possible to sort of look at the world around us at the genetic level using this genomic technology.
You're going to talk at all about the phylogeny, Nels?
Of course. So that's like when you can't see something, what do? You just keep looking at sequences and comparing it to others. And that's
tree is amazing that they put together.
Do. Yeah. Okay. So we've stepped through this. We've talked about the 189 genes. They immediately kind of categorize it in the archaeal family based on these sort of marker genes that you find in a lot of archaea. But then what you want to do next as an evolutionist is compare them to other genes like that. And so I think they take a sampling of 70 of the genes that you find. It's time to build a tree. You take 70 of these genes, put them all together, sample that from something like 150 different species of archaea, and then from different groups that have there's been sort of a gold rush in that space over the last decade or so. The Asgards that have been taken from the deep vents and sort of redefined the tree of life. There's been a ton of metagenomics in this space. So you kind of know the family tree is sort of filled in some of the gaps there. And so what's nice is they can take advantage of that to put Succinarium into the mix here. And you're right, Vincent. So when they do this, it's a little bit wild. So
this is amazing.
Yeah, pull that up. So there's that red branch. So we're looking at the color coding here are the 150 or so comparisons, the branches on the tree, or it's more of a phylogenetic bush than a tree, the way they've
these are
drawn
different. These are different archaea, right?
Yeah, exactly.
In the main part of the different colors, the halo, thermo, etc. Right?
Yeah, exactly right. And so you might think, at first approximation, that this one would just fall into one of those 3 or 4 main categories, but it doesn't. So it's sitting out there on this long branch, that red one, that's Succinarchium. And immediately as a phylogeneticist, when you see a long branch that sort of doesn't really go into a place with high confidence, and that's the case for their data. So they do, a number of different ways of drawing trees. There's maximum likelihood and Bayesian statistical approaches to try to get some confidence about the location of, or the relationships between these groupings or clades of different species. And when they do that, they just don't have a lot of confidence. And that's not too surprising when you have something that's just sort of new or out of the blue. But you have to be cautious because, so it turns out, that we draw these trees, a tree like this, even for ones of, say, like primates, our own closest cousins, it's a hypothesis. It's an idea based on the information that you decide to compare, but it doesn't really capture evolution as it really happened. We would need a time machine to pull that off. And so just with their different kind of techniques that they use or statistical tests, they point out that they could put that branch sort of in 16 different places. They would, it wouldn't be, associated with those groups. It would still be sort of next to them, but their confidence here is fairly low. And then it's even worse, Vincent, because it's a long branch. And so there's this really well-known issue of something called long branch attraction. When you have something that's just so different in a tree, how it kind of shows up, the way phylogenetics is sort of interpreting these relationships can get fooled by these long branches. And so things that are actually not related sort of associate with each other based on just sort of an artifact of the way that the analysis
but
is
the length
done.
of that means it's very distantly related to all the other sequences that are in the bush, right?
Absolutely. And so that's pretty clear. And then they, to their credit, they go in and they do sort of a couple of different flavors of analysis where they really try to account for that, the rapid evolution, the differences. And when they do that, they sort of get now a weak association with one of the groups. It might have been that one in yellow, the Nanabdelati, which is one of the major groups of Archaea. But even then, it's still very just sort of on the edge of its association with that, and the statistical support is pretty low. And so, they kind of go through all of this work trying to account for these things, and then in the end, they sort of, concede that the definitive phylogenetic position remains elusive. I'm kind of quoting from the preprint now. But that the succinarium represents a previously unknown branch, potentially warranting classification at a higher taxonomic rank. It could be a new phylum, or even higher. And so when we think about taxonomy, to sort of, an entire kind of branch of life to sort of show up, today is a pretty major advance potentially here, with all of sort of the caveats that we just discussed.
Just goes to show we haven't seen it all yet.
No, I know, it's incredible. Like the amount of life that's out there. We're kind of still at the tip of the iceberg stage, especially in the microbial world, in the oceans, the deep oceans. And so that's why, all of these discoveries in the last few years have caught so much attention from evolutionary biologists and beyond, as we're still grappling with what is life around us look like. So, but now that they have this foothold of this one new genome, the good news is you can now use that to search all of the other data that's out there. And in particular, there's a lot of great public data sets as part of the Tara Oceans project. And so they take advantage of that, and I think a couple of other data sets to look for other sequences that might match that, and to see, so that's actually, to step back, I think it's important to say that you don't want to get too far, too excited about one sighting. Is that, just with the artifacts that are possible with genomics, including at the single cell level. And so immediately you want to go into the other databases and ask, are there other sort of examples, relatives out there that have been also sort of hiding in plain sight? And the answer appears to be yes. So very quickly, by sort of filtering through or looking for matches for some of the sequences in the chromosome that they assembled, they find matching archaeal ribosomal RNA in those data sets. And so that is a kind of a nice advance, because now it's not just that one branch on the tree, but it's sort of now hinting at its own clade. so that there's other sequences out there. This isn't sort of a one-off where you've just, you built the libraries of your to sequence, to deep sequence, and stuff just got kind of mismatched together or something like that. It looks like there actually are other related sequences out there. What's really fun.
These are found all around the world because the Tara Oceans expedition, they find it in that data set, right?
Yeah, exactly. And so we're going in the same direction here. What's really fun is you can take exactly that kind of data, the sort of in a couple of different ways. So the geographic location, and so they look for where are those sequences, where were they collected, and it looks like they're highly enriched off the east coast of South America, the coast of Brazil, and so they're not found all over the world, but they're sort of in these areas, and the same dinoflagellates, the hosts, are known to be there, and so that kind of puts the right kind of building that circumstantial case. It's sort of the right host with the right archaean, and then they also can look at this by how much they filter those samples, right? So you're collecting all of the seawater or ocean water and then putting it through filters and sequencing different sort of cuts of the filtration. So you take stuff that's really small, 5 microns and under. The next cut is some 5 microns to 20 microns and 20 microns and over. And so they find those sequences enriched in the 5 to 20 micron range. Now, this tiny archaean, which we still haven't seen any microscopic evidence of or physical evidence of, just genomic evidence, but is much smaller than that. And so that also is consistent with the idea that it has a host. Now, here's where, things don't completely add up. So those dinoflagellate hosts that they started with are actually closer to, I think, 40 or 50 microns. And so they're too big and for that. And so how that all sort of relates to the relatives, who are the hosts exactly, is still a very open question. But it's kind of fun to do this like genomic dumpster diving and all of the publicly available data sets. So just by getting a sequence to try to learn a lot more about it, where does it live, which, and what are its potential hosts based on some of these features. So then back to the genome, Vincent. So they note that what's in there, what's among these 190 genes or so. It's enriched for the so-called GIP functions, GIP functions, genetic information processing.
The first for me, I never heard of that.
Yeah.
I guess it's a KEGG category.
I think that's right. Yeah.
Okay.
That's sort of a theme, I would say, of metagenomics, is categorizing stuff and having acronyms that you have to sort of wade through, right? And so this is one, genetic information processing, and that's just pointing out that sort of the things that are involved in DNA replication, RNA production or transcription, and then the ribosomal components, or the protein translation from RNA to protein. And this is, we kind of hinted at this already, but this is very different than a lot of the bacterial symbionts that have all these metabolic functions, and really sort of, in some cases, complicated, almost like Russian doll configurations. And there we have like really beautiful EMs of some of the bacterial symbionts, and how they associate in these sort of specialized organelles in the host, and then how the metabolism sort of might follow in some of that compartmentalization that can sort of evolve in these kinds of complex relationships over long periods of time. So if it's true that this thing does not encode those functions, and we sort of already covered that idea, that still seems like an open question. But then this is what points to this more sort of potentially parasitic lifestyle or replication style that sort of approaches a little bit more of how we think about viruses, right? And so that's where I think this paper, why are we into our more than a half hour now of kicking around the details here, is it sort of opens up that idea in the sort of the symbiosis field in a way that I don't know has sort of been considered before, or even this kind of circumstantial evidence has sort of come forward. And so I really like that, because it sort of opens up a new way of thinking about the transitions of life forms. I have to confess, in my own work, Vincent, I've sort of been thinking about these genetic entities sort of going in the other direction. So you and I have been together at these giant virus meetings. And so these are clearly viruses. So these are, need hosts to replicate, have all of the sort of fit neat, sometimes neatly, into these categories of sort of true viruses. But they have, their genomes are massive, much bigger than this one, for example, 5 times, 10 times bigger than this. They encode, in some cases, more than 1,000 genes. And so they have the genomic real estate to actually go from the viral or parasitic or symbiotic sort of world into something free living. And so I've been kind of toying with the idea over many years now of using the sort of techniques of synthetic biology. Can we tip them over, and can we kind of have genetic traffic go in the other direction where a giant virus becomes a free living life form? They sort of, you add all of the components you need for the ribosome, et cetera, and then see it sort of become life. Here, what they're inferring is it's going in the opposite direction, that you have a free living life form that's then letting go of that independence in order to be potentially more viral.
Well, and it's one of the two theories for the emergence of viruses, right? One, they started as simple replicons and then acquired genes when they went into cells. Or they started big, maybe cells spun off big viruses, and then they underwent gene reduction to get to a smaller place. And then some of them went really far, and maybe this is on the way, right? Because it has only some transcription, translation, and DNA replication genes. And even for translation, you only have 2 ribosomal genes. They can't make a ribosome, right?
No, that's right. Yep.
So they either acquired them because it helps in some way. Same with the tRNAs. They have 20 some odd tRNAs. Or they're on the way of losing them. And that's, you can't really tell which is the one here. But the thing is, this is not a virus, right? Because as they say here, it still depends. It depends on the host cell entirely. It really can't do translation itself. It can't do any metabolism. So it's I'm sorry, it is not a virus because it has these ribosomal RNAs.
Correct. It's the opposite. It still has the ability to to be alive, to do this without a host.
We don't even know. Because
we don't know.
We've never seen this. So Nels, if you took this circular DNA and you injected it into a dinoflagellate, what would happen?
No, I know. So so probably nothing, but because there's a lot of technical hurdles there. But that, I hope that they kind of are trying that, or sort of collaborating.
Actually,
to do that.
Maybe it is a virus. Now, I mean, the ribosomal RNA puts it into the archaea, right? But there are some viruses that encode a lot of tRNAs and a lot of ribosomal RNAs as well.
I know. Yes,
but they're eukaryotic ribosomal RNAs. They're
correct.
They're not archaeal, right?
Yeah. And there are viruses of archaea, sort of the equivalent of phage. So I think, my hunch is that kind of all of this is possible, and given enough time, and we have quite a bit of time when we think about 4 billion-ish years, plus or minus, for all of these things to potentially happen. Kind of genetic traffic in both directions, becoming, independent or free life forms from viral, the reverse, free life forms sort of spinning off parasites, viruses, symbionts, kind of all of the above. And so I think all that's possible. What's interesting about this is it sort of shows the potential of that kind of genetic traffic going in a different direction, or sort of from a different street or a different route. It opens that up. But getting back to your point, Vincent, that's exactly, I think, as we're doing our podcast peer review here, you want to see more evidence, sort of corroborating evidence, or evidence coming from sort of different techniques. So the most obvious is microscopic. What does this look like as can you see this thing? So I think that's where the connection between the coast of Brazil and the Terra's Ocean Project, where they actually see other sequences like this. If you are in the business, I'd send out a boat and collect start collecting seawater exactly where they had those, to see if you can actually do what happened with the Asgard archaea when they went to the deep sea vents. Can you actually sample this and do kind of a microscopic examination and link that to the genetics that you see here? Again, easier said than done. The oceans are massive, and I don't know the resolution, sort of the geographical resolution of those data sets. But certainly, at least it sort of narrows your search space to some degree. And of course, you're sort of hoping that this is like an abundant life form, and I don't have a sense of that. from the work so far. But then your point is a really good one. And given the proposal that this is that this is all the kind of genetics that you need on this circular kind of chromosome, 238,000 base pairs, you can make that. Synthetic biology allows you to do that. And then you could try to launch it in the exact host that you've been looking at. And so they have access to that. So that's a relatively accessible route. There's other technical reasons you could imagine it doesn't launch, because it doesn't have it doesn't have the sort of cell membranes and machinery around there that would allow it to persist or to sort of fire up. But that's possible. And there's certainly a lot of cases where synthetic biology has approached that in terms of some bacteria and certainly viruses that you can launch from sort of in vitro transcription in some cases, and you put it into the host. But then if they're missing plasmids, that would certainly go off the rails for biological reasons, but it could also go off the rails sort of for technical reasons. And so
but you have to just try it, Nels.
Of course. And I'm guessing that is happening. It's really interesting though, so like, as we look at the preprint, you can see it's sort of a polished presentation that, they're trying to kind of make the provocative case that this sort of changes the way we think about cellular life. And so I can imagine that they're sending this or already have, it might be under consideration at a high profile journal magazine. But is that as a reviewers, I'm curious your opinion, Vincent, like if you were a reviewer of this manuscript, would you want to see some more of that evidence, or is there enough here already that you think that this is, something that you would want kind of published in a high profile place to sort of get it in front of other colleagues, peers, et cetera?
I think this would be a current biology paper.
Mm-hmm.
I don't think cell science or nature. Okay. But current biology is very good, right? Because there are a lot of things missing here, and there's no paradigm established yet. Okay. This is the smallest, archaeal genome. But what if there are tons of plasmids in it that would make up for all this metabolic stuff, right? So as you say, that we don't know that. So I think to put it out there as something really different needs to wait a bit. I'm hung up on the virus thing. In the graphical abstract, they draw it like a virus. They have glycoproteins. It's a spherical particle with glycoproteins.
Yeah.
These are these big transmembrane proteins that they talk about. Yep. So, but yet in the discussion, they say, okay, so this entity has characteristics that are reminiscent of viruses, but it remains fundamentally cellular. And I think this is wrong. They write, a key distinction from viruses, which typically lack their own core replication machine genes and rely on host systems. That's not true. The big viruses have their own core transcription, and especially the cytoplasmic replicating viruses, right? The pox viruses, they encode a DNA synthesis machinery and a transcriptional machinery because they're away from the nucleus.
Yep.
So I don't know why they say that. That has to come out because that is not a key distinction. Just because this entity encodes DNA replication machinery, some transcriptional machinery, and a little bit of a translational machinery, that doesn't make it cellular to me. I think this has a lot of viral characteristics. So now tell me, why do they think it's cellular? By the ribosomal RNA homology?
Yeah, I think that's right. So as you go through that category of, I'm already forgetting the acronym, or these RIPS, GIPS, the genetic, the genetic information processing categories, to see that full list, where. Right. And then to infer that sort of that it's into that transition. So the argument, I think, leans pretty heavily on the idea that, some of those large genes are in that, among the 190 genes that they discover, characterize, that those aren't that those are all sort of moving towards that parasitic, non sort of symbiotic, bacterial symbiotic sort of pathway. And I think I think for me as well, for that reason, it falls a little short, just because that feels like it could be right there, actually, just sort of, because we don't know. But I don't want to cast too much cold water, because I think it really does open up kind of a way of thinking that still on thin evidence, but that really sort of shakes awake the field in a sense, to not just go down that pathway of the bacterial symbionts, which are sort of have dominated for decades for good reasons. But that's the excitement, I think, of seeing this new thing, is it just sort of opens up your thinking to what might be possible. But yeah, early stages here. And so in some sense, I think, having it kicked around and in front of as many scientifically curious human brains as possible makes a lot of sense, including our listeners here on TWiEVO. But still pretty early days in terms of how strong conclusions can you build from this. And so I think they kind of probably are a little on the speculative ground, is how I fall.
So Nels, there's two ribosomal RNA genes here, and it's on that basis that they say it's cellular. But as I said, there are other viruses, giant viruses like Tupan viruses that have ribosomal RNAs, and those mimic the host. They're eukaryotic ribosomal sequences. So if this is a virus of archaea, its ribosomal RNAs are going to mimic the archaea. So it's not evidence for cellular.
Correct. Well, and it can go, agreed. And it can go in both directions, right? So, so one idea, maybe a simpler idea, is that, so the reason that some of those giant viruses have some of these ribosomal proteins is that it allows them to sort of modify the translational machinery to their advantage, right? So as the parasites are kind of conducting their viral replication cycles in the host, puts up defenses to it.
Right.
That's sort of like the ribosome is definitely, sort of vied for to get to, basically to have to take energy from the environment and then, translate that literally into sort of replication, of harnessing that energy to use it to replicate. And so that's a pretty easy explanation that, again, with actually limited evidence, though, I would say. I guess, the sort of null hypothesis is that there's just a lot of genetic exchange and just sort of slop, where there's all of these sort of genes or RNAs are just sort of being exchanged in different compartments that then become viruses or free living animals, or organisms, I should say. And that we're just seeing kind of the genetic noise as opposed to the signal of something that's actually beneficial. But I think that's where I think it can go in both directions. Likely this sort of opens up the other conduit in a way that I think hasn't really been glimpsed at before. It could be that a lot of those giant viruses, maybe, so under their model, it's possible that they could have, kind of taken this route and are now have let go of their independent lifestyle, as opposed to this one seems like it's a little bit closer on that transition zone. But I don't know. We need our science buddy Eugene Koonin.
I think he would have a field day with this. Right? So look at the graphical abstract. They have this thing attached to a host cell. It seems to have fused with it, and metabolites are going in. But they're implying that this thing is carrying out translation on its own. It can't with only two ribosomal RNA genes. It cannot do translation. It would have to use that of the host cell.
That's right.
I'm just not convinced that this is a cell in any way.
Yep.
But Nels, they draw it like a virus particle, don't they?
They do. So that's where they've definitely kind of polished their story in a way that might be getting a little ahead of the game based on the sort of the genomic evidence that's in front of us so far.
I think it's very interesting. And this is a preprint, so I suspect this will undergo substantial revision before it's published. So we'll see.
Yeah, which is really fun to also watch that kind of in plain sight. That's one of the fun things I would say about kind of science publishing these days, is that it's very common now for people to put out sort of an early version like this, which is great because then we can pick it up, kick it around, compare notes. In the science, sort of the process, science advances a lot faster. In the meantime, kind of in increasingly transparent ways, but there's a lot of work to be done there. Peer review happens. And so it gets, people look at this who, their day job is to think about these kind of things and to critically analyze this and sort of come to an opinion with editors on what we're looking at here. So that process is probably well underway in a more kind of behind closed doors situation.
So your dad asks, genomic dumpster dive, is this a new discipline?
Yeah, we've been up to this in my lab for a while. So we, discovered a few years back a virus of zebrafish. And once we got the sequence, we could go dumpster diving. And zebrafish are especially a rich sort of, or a full dumpster, I guess, in this analogy. Because for 50 years, developmental biologists have been posting sequences from zebrafish, and sort of hiding out right in those sequences are viruses that were, not really seen before. And so just by getting that first, and that's what they do in this paper, I think really beautifully, just getting that first sort of glimpse or putting together that genome, then or chromosome, circular piece of DNA, they can then go to the massive database, dumpster dive, and you, without even picking up a pipette or doing any virology or whatever it is, you can learn a lot more just about where you find it, where, what are the relationship between other sequences out there. Biological dumpster diving is alive and well.
So Tom wants to know, so Eugene Koenig has these ideas, there's capsid encoding life forms versus ribosome encoding. Yep. Tough reading, but seems relevant. Yes. Right.
Oh, agreed. Yeah, completely agree. And I, with you on the tough reading part. It gets complicated in a hurry, and the sort of, I think it's like builds really nicely from sort of historical evolutionary biology, where the sort of sophistications of the arguments that are made, sometimes with very limited experimental evidence. And that's not to cast aspersions. That's sort of the history of the field before kind of the molecular biology era. And so anytime I've sort of approached some of this very interesting, but also very dense sort of argumentation or literature, I sort of shy away from it in the sense that I grew up, as an experimentalist, and sort of trained with the idea that we're looking, how do we simplify things as opposed to make them even sort of more dense or complicated. I'm not saying one is right or wrong. I mean, it's the history of evolution is what it is, and it's probably quite complicated. And so it makes sense that this is tough reading. And I'm glad there are people like Eugene Kuhn and others who are really, sort of tackling this, and sort of the daringness that it takes to build these arguments, to really understand this at this level. Takes some really big brain thinking, to say the least. But that's where, the confluence of experimental data, in addition to sort of piecing together these really complicated, deep origin kind of story sort of scenarios, I think that's the hope for the field moving forward into the future.
So Barb Mack notes that a dumpster in the UK is called a skip.
Skip diving.
Skip diving. It doesn't have quite the same.
Yeah. Hopefully not a dumpster fire, which is another situation here, right? As we're trying to discern biological signal from noise, you could find yourself with a dumpster fire pretty quickly.
So Pete wants to know, what's the largest genome you could synthesize?
Yeah. So I don't have at my fingertips the exact number, but this is, who's the guy who's used to run Tiger, the Genomic Institute?
His
name. Yeah, Venter, right? So he, in the synthetic biology field, they've launched some bacteria where they have the whole genome, but they make it from scratch, and then sort of launch it that way. It's much bigger than this, no question about that.
So millions, you could do a million gene bases easily, or
I don't know about easily, but you can, yes, you can approach that. It takes a pretty big technical effort to do that, but it's becoming easier and easier as.
So he made a 4 million base pair genome, Venter.
Yeah. Yep, there you go.
But I know that so before that it was 500,000 base pairs synthesized also by Venter. It was a mycobacterial genome. Yep. So you can get pretty big.
Yeah, and here this is lower than even that first one. This is at 238,000. So well under the sort of technological limit there.
All right, one more here before we do some picks. Daniel says, it's beautiful. It's like complex systems following specific rules inside more complex systems following the same rules, but like another exponent. It suggests a refined organization.
Yeah, and then I agree. There's like that kind of balance between the constraints or the rules, and then the chaos that just sort of describes biology, all sort of mingling. It's really, really fascinating and worth thinking about.
TB wants to know, why are there no viruses of mitochondria? There are
That's right. Yeah,
mitoviruses, and they're thought to have come in when the bacteria came into the cell. It brought its viruses with it, and they're still there, mitoviruses, right?
exactly right. Yeah, and so when we think about origins of viruses, we don't have to go to these sort of complicated. You get to a sort of complex life form or cellular life form, and then sort of strip it down. Um, Also, even in the kind of RNA world, before DNA, sort of the dominant genetic code. There's, I think, good ideas and good evidence that
Mm-hmm.
kind of parasitizing on things around you, or cooperating, becoming addicted, taking advantage of all of those things were happening early and often. And that kind of mirrors a lot of the thinking with a lot of the streamlined genomes, like small RNA viruses, et cetera, where it would come up from that very ancient origin. But that's, again, I think this is either or. It's sort of all of the above, all of these natural experiments that are, possible and sort of overlapping, all happening at the same time. Potentially kind of modern, like in a mud puddle next to your house, there could be a virus that just got the last gene it needed to become a free life form, and there's a new origin of life right under our nose. This could have happened multiple times,
Speaking of mud puddles next to your house, Nails, you have one that you've been studying,
yeah.
right?
Absolutely, yeah. We're all about the aquatic systems these days. Yeah.
So
Right.
Pete says, so pox virus could be constructed. Yeah, it's been done. Horsepox virus is extinct, and they recreated it with synthetic DNA. Yeah.
That's right. To some controversy, because that sort of, as that was published, you could imagine pretty easily moving that over to a pandemic causing pox virus. Someone could use that as a, bioterrorism sort of scenario. Yep.
Well, the thing is, we have to be prepared for that, right?
Absolutely. This is a time to rededicate ourselves to understanding viruses, where they come from, how they evolve, adapt, what are the sort of evolutionary potential for new pandemics to emerge, and how do we counter that? How do we have the sort of infrastructure in place to do better than we've done in pandemics of the past? And unfortunately, as we transition, I think, from the science probably back into sort of the science situation in the U.S., given current politics, et cetera, really dangerous days where we're going in potentially in the wrong direction, inviting viruses back into our sort of public spheres as we potentially dismantle or already have dismantled the vaccine committees at the CDC, as RFK Jr. and others are sort of burnishing their own agendas here. Feels like really dangerous days where we're potentially moving backwards instead of forwards in our pursuit of knowledge, in our pursuit of new strategies for promoting human health versus, kind of snake oil interpretations of it.
They canceled this consortium that was looking for potential pandemic viruses, right? I mean, they said it was too dangerous to collect viruses. That's ridiculous.
Yeah, kind of the ostrich putting its head in the sand sort of impulses here at the moment in U.S. politics, which are really, really troubling. And I guess maybe, Vincent, that nicely transitions to our science picks of the week. So I'll kick it off here with the Bethesda Declaration. So we'll have a link here. So if you go, before we get the link up, if you go to Stand Up for Science, or just in Google type in Bethesda Declaration, this is a document that just came out. Still looking for signatures. So it's this is a letter to the current head of the National Institutes of Health in the U.S. And the title of it, it's a call for NIH, the National Institutes of Health, and HHS, Human Health Services leadership, to deliver on promises of academic freedom and scientific excellence. And so I think it's pretty nicely summarized. It's relatively short, but it outlines, these are some NIH employees who really stood up in this moment, I think, effectively made a very clear argument and case for what's happening all around us here, raising these concerns about the how science is being newly politicized, or politicized in new directions, which is impeding or halting high quality peer reviewed grants and contracts. This is impacting health disparities research, work, as we're discussing with viruses, COVID-19, immunization, halting work on the impacts of climate change, gender identity, sexual health, and broad participation in biomedical research. And in fact, we just saw in the last couple of days here, I think a judge who's been on the bench for 40 plus years, appointed by Ronald Reagan, who reversed some of the canceled NIH grants and noted in a very angry tone that the cancellations were highly discriminatory, just sort of, clearly violating U.S. law. And so hopefully, we're starting to see some counterpunches here. But anyway, the Bethesda Declaration outlines some of these exact issues. And so urging the restoration of grants, urging the restoration of global collaboration. This has been the latest attack, or one of the latest attacks on NIH science, is that grants are being canceled because they have international collaborations, which just makes no sense at all in terms of how, as we talk about on TWiEVO constantly, and including the preprint today, science is an international game. It's not just people in the US that are practicing it. It's enmeshed across the world. The undermining of peer review, which is underway in many ways, as we're seeing delays in reviewing grants, massive backlog building on NIH grants, and then the cap, the 15% proposed cap, or an executed cap in some cases on indirect costs, which just throws a grenade into how we conduct science or organize our science at institutions, public institutions, private institutions across the nation. Mm-hmm. And this isn't to say, finally, and a really important point here as well that comes up, the firing of essential NIH staff. And this is both in sort of conducting peer review, but also the scientific staff, the intramural research that goes on at the NIH, which has been hobbled. And we get these like awful stories about how hard it is to conduct science under current conditions there. And so this is all illegal, the stuff that's happening as we're seeing some of these court cases being litigated or just sort of plainly violating a lot of this, a lot of not just the tradition, but just sort of the legal and constitutional constructs of how the U.S. is set up. So the Bethesda Declaration has been out for a week or two now. It's been signed by more than 27,000 people. If you haven't already, please go to the link, read it, decide if you want to sign on. I signed on a week or so ago, just trying to stand together and supporting NIH-funded science and to sort of return to something that resembles, or at least, I don't know that we're going back to science as it was practiced before, and science can use some reforms too. Don't get me wrong, but the attacks and the sort of snake oil that's really dominating right now is very troubling, to say the least.
Yep, I've signed it too.
Yeah.
I mean, you can't just say we're not going to fund any grant with DEI in it. That's just totally racist. Okay.
Yeah, illegal, discriminatory, and that was said by and the judge on the bench there, Reagan appointee, has never we saw this actually with birthright citizenship. I think another Reagan appointee, a judge on the West Coast, who are just, as this goes through the legal process, just baldly illegal and reprehensible, honestly, as well. It's just,
as you know, Nels, this administration feels it's above the law. No king, Nels, no kings.
No kings. A new national holiday, June 14th, King's Day.
Established this country to get away from the king, and we're not going to let the king back again.
No, and that's part of our agenda here on TWiEVO, is to talk about this, to lift up these issues, and to try to enlarge that conversation one way or another. Speaking of science in the U.S., Vincent, I think I could use a drink. What's your what's your science pick of the week?
That's exactly what my pick is. This is a Medscape article by Emma Rumney and Jessica DiNapoli. It is called U.S. to drop guidance to limit alcohol to 1 or 2 drinks per day, sources say. So the U.S. dietary guidelines are expected to be revised. Currently, they say adults should not have more than 1 or 2 drinks a day. It's a recommendation, okay? And now they're saying, because in part the pressure from the alcohol industry, they want to
Oh
get
yeah,
rid of it and
yeah,
make no recommendations. Okay. And this just reminds me of tobacco, right? Well,
yeah.
it's causing cancer and you don't
Hmm.
want to have it regulated. You don't want the information out, right? These nasty companies who are interested more in profit than in people's well being. So I think this is disgusting. So the UK has guidelines limiting drinking to 14 units per week. Units. Well, a pint is huge, man. You could have 14 pints. Anyway, it makes you, I want you to just think about this, right? Because there are health risks of excessive drinking for sure, right? And I don't know exactly what, RFK Jr., by the way, has been completely silent on this. He doesn't drink, which is
Yeah.
fine.
Yeah.
But why is he silent on it? He wants to make America healthy again. Speak up. Anyway, so there are health problems associated with drinking, and I don't think it's a bad thing to say you should only have one or two drinks a week.
Oh yeah.
You don't have to make people here. You go to a party, you can do what you want. But I think that having the industry, they have lobbied millions of dollars to get this changed, which is disgusting. I hate lobbyists and lobbying. I think this is disgusting. You pay Congress to do what you want. This is awful. So I think you should think about this. And I mean, this is the U.S. Dietary guidelines. It's one of the many changes that's happening in U.S. public health, and
Yep.
you know,
No, I'm with you. I think, and your point about guidelines versus sort of policies, right? And so, and which is also a changing as more information comes in and is digested and sort of then changed into guidelines, allowing people to have the best information we can get to. That's going to be updated as more information comes in. Very difficult to really pinpoint the exact. It's complicated, right? Human physiology as it relates to alcohol consumption, the exact impacts at the population levels, but to do the best we can and then let people make decisions as they go forward. I mean, I have to say, I'm personally in a little bit of a renaissance right now with the fake beer industry has gotten so good. Like the zero alcohol beers have really come a long way. This has been a game changer for me in
yeah,
terms of
they
as
taste
I'm
good.
yeah,
Really?
there's some great ones out there. Maybe we've skipped the ads, but I won't do any sort of sales pitches here. But really, so, yeah, some great advances there. And at the same time, obviously, alcohol is a big part of just human interactions as well, and in both positive and negative ways, and everything in between. So it gets complicated. But I think the troubling sort of notion, or what I'm seeing the trouble, and kind of stepping back and looking at the bigger picture here, is how these issues are sort of like as now we think about current leadership, for example, at Health and Human Services, RFK Jr., et cetera. Sort of, just that kind of making the case when it's to the advantage of the person, as opposed to sort of that bigger picture. What's the information and how can people use that in their own situations?
Yeah,
And so,
yeah.
I think historically on the right of politics, people have said, oh, well, you kind of liberals are all telling us what to do with these guidelines, et cetera. Like when Michelle Obama launched her, healthy foods for like elementary school students, there was massive pushback on the right. Now, flash forward a few years, decade or whatever, and we've got RFK Jr., crusading against artificial dyes and things like this, but not just sort of suggesting it, like literally trying to change policy, wiping out vaccine boards. I mean, the vaccines that may be available, or it's up in the air right now. So this isn't just guidelines or recommendations. This is potentially like changing policies in what the right, I think, historically would call a massive governmental overreach. And yet here we are in kind of snake oil land. It's the mismatch here is really wild. And so I think that the challenge is for like scientists, others, public health voices to draw that distinction and sort of the hypocrisy that's coming out here and the difference between, what is a guideline or a program that's looking to promote health versus sort of the new sheriff in town kind of energy that we're seeing with RFK Jr., which is really troubling, especially as it relates to vaccines. That feels to me like since our last podcast, even, right, that feels like that's moved in a really negative direction really quickly here, as we're all sort of holding our breath, honestly, for the next flu, COVID, et cetera, season, and what's available based on the misinformation that's being sort of weaponized in real ways.
So the ACIP, which he has reconstituted with bogus members, is meeting next week, and the agenda includes a discussion of measles, mumps, rubella vaccine. So they could withdraw the recommendation for that for kids. They could withdraw, and then nobody would get immunized with MMR anymore. Then you have huge outbreaks of measles in this country. So this is unacceptable, folks.
Yep.
We can let's wrap up with a couple of comments here.
Good idea.
Come in just a few. Here's one from ZZ Captain. Hey, you remember saying that nature's by far smarter than we are? Yeah, sure. Take me a long way in science health writing. I definitely agree with you, and thank you for that.
Mm-hmm.
Well, I didn't think of it, that's for sure. But yeah, nature is a better everything than we are.
Well, it's had more time, right? So like billions of years.
But even Nels, you have you have an oil spill, and within months the bacteria change and respond to it. It's amazing how resilient it can be.
All the natural experiments, tapping into that sort of natural wisdom is a real thing. Yeah, and echoes through evolutionary biology for sure.
Daniel is politicized in an absurd direction. Scientific research and robust information are being ignored, and it's obviously dangerous to everyone. Totally agree. Mm-hmm. Totally agree with that. Pete tells us a pint of beer is 2 or 3 units. Okay. So 14 units a week. Okay, got that.
Yeah, I was wondering if your conversion might be off by a little bit there.
14 units. I didn't know what a unit is. A UK unit is 10 mils of alcohol. Alcohol, though, that's different from a drink, right?
Yeah.
Let's see here. ZZ is allergic to alcohol. Well, there you go. You don't have to worry about recommendations, right?
Yeah. Try the fake beers. Although they still, I guess they, as they're brewed and then the alcohol is removed, I think in some cases it sits at like 0.5% alcohol or a little under for a lot of these. And so that still could be a
number of people are allergic to that. So Barb Mack gets histamine reactions, and John gets flushed. Now I drink soda water. Yeah, try non-alcoholic beer, John. You might like to do that. And finally, Tom says RFK Jr. and credentialed cranks. That's right. That's a good word for them. But, their credentials are all in the wrong areas, not in vaccine science.
Well, yeah, and some of those credentials actually are cooked. Like, so they're using AI to even in some cases to, like summarize the credentials of some of the people, for example, on that ACIP board. AI gave one of the members a PhD. I think it's an MD, who now is listed as an MD PhD because AI just sort of hallucinated sort of an extra credential there. And so the stuff they're putting out there ain't real.
All right, folks, that'll do it for TWiEVO 115. Let me thank our moderators for being here today. We had Barb Mack, we had Les, we had Tom Steinberg, and Vanity Nutrition. Thank you for being here and making it civilized. And thanks all of you for being here. We appreciate your coming. We'll be back in about a month. You can just check the schedule at microbe.tv/calendar to find out exactly when. So TWiEVO, show notes are at microbe.tv/TWiEVO. Really, if you can, please support our work, microbe.tv/contribute. Nels Elde's at cellvolution.org and on Blue Sky Early Bird. Thank you, Nels.
Yeah, thank you, Vincent. Good to be together and looking forward to TWiEVO 116, probably coming out in about a month or so.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on TWiEVO is by Trampled by Turtles. Let's get that music started here. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, a podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious. Bye-bye.